All right, happy lunchtime. My name is Ollmann. I'm the Director of Philanthropy and Social Impact for the Jewish Community Foundation here in San Diego. Thank you for signing in for the first in our online series called Access to Insight. We know this is a truly unprecedented time in all our lives, and we're all having to readjust in terms of what it means to be connected to one another. Right now, I'm not talking to you from the Jewish Community Foundation offices, but instead from the room in our house that will hopefully be our baby's room come August. This is my makeshift home office for now. I know that every one of you is having to make real changes in your lives, and we know many people are experiencing real challenges. The goal of JCF's Access to Insight series, of which this is the first, is to help reestablish a sense of connection and of community. And we want to do that with some real purpose to it. That purpose is to educate and help inform you of what's going on around us, and to do that by bringing speakers to you with incredible first-hand information and resources. Now, I see that there are 125 people on this call right now, which is pretty amazing. Many of you are familiar with the Jewish Community Foundation, but some of you are not. So before we turn the call over to Dr. Saphire, I just wanted to share briefly about the Jewish Community Foundation, who we are, and what we're doing at this critical time. So the Jewish Community Foundation partners with individuals, families, and community organizations to help them achieve their philanthropic goals. Since 1967, we've facilitated over $1.5 billion in philanthropic grants into our community. And I'm just going to put a slide up so you can see our professional leadership team. All of who are on today's call. But I also want to acknowledge our amazing lay leadership team led by JCF Board Chair Janet Ashertel. The Jewish Community Foundation believes that every individual has the power to make a difference, and that together we can change the world. Our mission is to promote philanthropy through meaningful partnerships with donors and community organizations to achieve their charitable goals, and to increase current and future support for a vibrant and secure community in San Diego, Israel, and around the world. And never has that mission been more important than right now. As a primary trusted and expert resource for philanthropy, the JCF continues to educate, engage, and inspire generations of givers. Today is one of those special opportunities to educate, engage, and inspire. The La Jolla Institute for Immunology is a 501c3 nonprofit working on behalf of all of us right here in our own backyard. Just 5 weeks ago, we took some JCF donors. I can see Dr. Saphire laughing, because we took JCF donors on one of our regularly scheduled educational site visits to the La Jolla Institute. We heard from several incredible speakers, including Dr. Erica Ollmann Saphire. She has a very impressive background, which I encourage you to review later at the La Jolla Institute's website. She's an expert on viruses and diseases at the molecular level, with expertise in Ebola, Lassa, and other viruses. She presented about something that many of us had heard about at that time in China, called this novel coronavirus, COVID-19, and the role that the La Jolla Institute was playing in trying to understand and address it. Dr. Saphire blew us away with her presentation, but none of us, I'm sure including Dr. Saphire, had any idea how in such a short time it would fundamentally change our lives. We're so grateful to Chris Lee from the La Jolla Institute for arranging this, and of course to Dr. Saphire for being willing to give this update to us. Now I'm about to turn this over to Dr. Saphire. Before I do, let me tell you how this webinar will work. Dr. Saphire is going to present to you from her lab through the magic of Zoom. After that, there'll be some Q&A. And then after that, we will spend a few minutes explaining how JCF is supporting the La Jolla Institute and many other organizations as they address many different aspects of the societal impact of COVID-19. You can submit a question within Zoom by finding the Q&A button. Just roll your mouse over the screen or touch the screen on your phone and tablet, and then just hit the Q&A button and hit submit. you can do that at any time starting now. And if you run into any technical problems, you can email Mara, M-A-R-A, at jcfsandiego.org. So once again, Dr. Erica Saphire, thank you so much for being here and presenting to our community, and I will turn it over to you.
It's such a pleasure to see you, and I wish I could see the rest of you. I always prefer to have people in person, but here we are. I can show you a little bit about what our laboratory is doing and tell you and help interpret some of the things that we've all been hearing on the news. So let me share my slides. There we are. Do you see our new friend? The virus there? Let me just try to move this. Okay. So it really wasn't very long ago, New Year's Eve, did not even 3 months ago, when a new virus came on the scientific consciousness, which we are now quite familiar with, the novel coronavirus. Now, this is one that you have had before. The coronaviruses typically cause the common cold. There are 4 that do that have these names here. And they just come and go seasonally, and they don't make anybody very sick. And they weren't even very interesting scientifically until SARS emerged, another coronavirus, in 2003. In fact, it was really hard to even get a research grant to study coronaviruses because they just sort of cause a small cold in humans until 2003. And they cause other things like mouse hepatitis virus, which was just a research model. The first really dangerous one we saw in 2003, and that was SARS, which emerged with cases linked to a wildlife market in China. The 2 intermediate hosts, one was a palm civet cat, this guy here, that was being sold for food, and that was the source of the spillover. When there's a new virus that emerges from an animal population, we call this a zoonotic transmission, zoo meaning animal. It's something that's new to humans that came from some animal host in nature. And it might have been a couple animal hosts. It might have been interactions with bats and civets. And it spills over into humans. So something humans haven't had before, something else in the wild. So that was SARS in 2003, which was about 10% lethal and infected 8,000 people. And then later, MERS virus emerged, the Middle East Respiratory Syndrome. And this one has multiple transmission events that even were ongoing fairly recently from camels into humans, also with a bat reservoir involved in there. And there are about 2,500 cases of MERS, serious, it's about 30% lethal, and there was an outbreak in South Korea from travelers that emerged from the Middle East. Now this new guy, the virus is called SARS-CoV-2, the disease is called COVID-19. So that's kind of like the virus called HIV and the disease called AIDS. That's the difference between the 2 words. The virus is SARS-CoV-2, the disease is COVID-19. It emerged in Wuhan, this place in sort of Central Eastern China, and was first noted among a cluster of people that worked or shopped in this seafood market here, where a lot of seafood and also mammals are sold for food in both legal and illegal ways, including some of the hosts that we know and have been associated with pandemic coronaviruses in the past. Now, when researchers and scientists went in to test, they looked at a lot of samples of different kinds of meat in this market, and a number of them were positive coronavirus. And so coronavirus definitely stayed in this market. It was closed, I think, January 1, and it propagated some of the start of the outbreak. But some of the first cases weren't associated with the market, something else. So there was some other ecological transmission of the virus before it spread in the market. And it could have spread in the market just because there's a lot of people clustered together. It could have been person to person. It could have been from the meat in the market. So SARS-CoV-2 emerged there. There are enough differences in it from viruses we've seen before that no human could have made this. This is a natural and wild virus. It is closest in similarity to something that has been observed in bats. A little piece of it looks like a coronavirus observed in pangolins, those funny looking armadillo-like creatures. So there could have been some recombination where it grabbed a bit of what we call the receptor binding domain from the pangolin virus and inserted it into the bat. But there are so many differences throughout the thing that this could not have been made by a human. This is a natural virus that just emerged from Mother Nature and the normal amounts of genetic drift and change that happened there. And so what we observed very quickly from the first few cases in China with the rapid and extensive global travel is how quickly it spread to other nations. And so this is January through February, which is very much the start of the outbreak. You can see how quickly cases are popping up in different places around the globe. And this is just what viruses do. We anticipate this every single year for flu, and the abundance of flu is tremendous. These were the case numbers last night. I like this website a lot, Johns Hopkins University's coronavirus resource. So 470,000 with 20,000 deaths, about 800 in the United States. And if you look at the cases that have been happening just in the US, you can see we're still on that upward trajectory. We're still climbing up that asymptote because so many of us are naive. And this is just what viruses do. And so if you look at what's going on in the rest of the world, so the orange graph is now China, rest of the world as of 2 weeks ago, yellow, and the recovered cases in green. Now look at the orange graph in China. It was a tremendous epidemic in China. It climbed up very rapidly. Then through fairly draconian social separations measures leveled off. And so the number of cumulative cases you see there in orange is pretty level. They're having very few new ones. And so that kind of shows that those kind of draconian measures of keeping people apart from each other are successful, that transmission stops as far as we can tell, and to the extent that we can see the data that really are. Okay, now the rest of the world, that was then. What's going to happen now? Probably something like this. I would expect China to stay relatively level with few new cases. The number of recovered cases, green, that's going to go way up. That's good news for everybody. Those people are now immune, and we're going to rely on that herd immunity. And what's going to happen for the rest of us in yellow? Well, it will certainly keep going up for a while. And at some point, it will level off and become more stable. We don't know yet when that point is going to be, but we are very much here on this upward climb. And so the concern is this virus on top of the normal amount of respiratory disease we have all the time, and needing to build that herd immunity slowly because we just don't know when it's going to level off. So why stay away from each other? It's because it's the only tool that is widely deployable right now, and that's a little bit sad. The vaccines will come on board, but not for a year or two. The drugs are being analyzed now. We're raising antibodies now. That's all going to take a couple of months. And so why social distancing? This virus transmits in two ways. We've learned a lot of words in the last couple of months. We've learned coronavirus. We've learned social distancing. You've also may have learned respiratory droplets, and another one, fomites. So respiratory droplets is exactly what you're seeing right there. When you sneeze, when you cough, even when you speak, small, sometimes visible, sometimes microscopic droplets of fluid come out of you, and they go forward about 3 to 6 feet. So if you're 6 feet away from anybody, the little droplets of fluid will not hit you. And that's good. So why does a virus transmit in little droplets of fluid? That virus has to stay moist. It's got kind of a membrane envelope, like an oil-water interface. It's really delicate. This virus is easily killed by soap and bleach and UV light and a couple of days on a dry surface. It will last a few days on something that it could make little molecular bonds to, like fabric or wool. It will die very rapidly on copper, last 24 hours on a hard surface. So you can spread it through respiratory droplets. You also spread it on those surfaces through the other word I used, fomites. That's what you touch something and then you touch your eyes or your nose or your mouth. Your skin is a remarkable immune protective organ. And although a virus might land on it, it does not infect you through that dry skin layer. Viruses get in where you're vulnerable. So if you've watched Star Wars, got kids that watch Star Wars, remember how Luke Skywalker had to fly in through that one hole in the Death Star to find that one vulnerable spot to get in there to shoot. Your eyes, your nose, your mouth, or any little cuts you have, those are the openings in your own personal Death Star that your skin is protecting. So if you touch a railing, a light switch, a shopping cart, anything at all, and then you touch your eyes, nose, or mouth, and we do this constantly, that's a way a virus can get in. So that's why they're saying to keep your hands away from your face. Okay. Now, how lethal is it? This is quite ambiguous, and the answer is that we do not really know, and we will not know until it's all over and we can do the serology. So let me walk you through this. So MERS is quite lethal, about 30%. SARS, also quite lethal, 10%. This new coronavirus, somewhere between 1 and 3. And the reason we don't know how lethal it is because we don't know how many cases there are. Because you see the sick ones, you do not see the asymptomatic cases. They're not being counted. So the estimate is 1 to 3, but why the range? And the range has to do with how many they're counting. So this data I'm showing you here is from the good old days of about 2 weeks ago, which now seems like forever ago in the course of this pandemic. And here were estimates of lethality at the time. So really early on, we only noticed the really sick people in America, and we came up with 8%. We know it's much lower now. So let's look at 3 countries here: South Korea, Italy, United States. Now, a case fatality ratio is a fraction. And a fraction has a number on the top and a number on the bottom. It has a numerator and has a denominator. What that case fatality ratio, that fraction, will look like has to do with how big the number on the bottom is. We see the deaths, they're very clear to us. We see the very sick people, they're very clear to us. We do not see the asymptomatic people. And we do not see a lot of the people that just kind of got some mild thing that they didn't think much of and cleared. And we don't have enough tests, and we haven't done enough testing to have found those people anyway. So what does it look like depending on your level of testing? So at that point in time, South Korea had 34 deaths of, all right, all these little faces are moving. I can't see my own numbers. Let me change this. 34 deaths over about 5,000 cases. And so they come up with a case fatality ratio of, I cannot see my own numbers. Much better. Okay, play again. They come up with a case fatality ratio of 0.7%. Now, if they had not done so many tests, they still would have found the 34 deaths, but they wouldn't have found as many cases. Let's say they'd only tested 1,000 cases, they would have come up with a case fatality ratio of 3%, and that's a whole lot worse than 0.7%. But if they had done a whole lot more tests, let's say they had found 15,000 people, including all of the ones who are asymptomatic that otherwise wouldn't have been noticed by anybody until they've been tested, they would have come up with a case fatality ratio of 0.2%. And so the appearance of that case fatality ratio solely has to do with how many people you have found and how many people you have tested. Now, our testing was an extremely slow and poor start. 300,000 people now have been tested out of our population in America of about 327 million. So we've tested about 1 in 1,000 people. Now, something you can expect to read in the headlines that you should be really rational about is that as testing ramps up, more of those mild and asymptomatic cases will be found. Now, we all know they're out there. There are probably 10 times as many as have been counted now. But once they are officially counted, they'll be added to the official numbers, and it will read in the headlines like a disaster. Cases shoot up tenfold or something like that. It doesn't mean the virus has suddenly gotten a whole lot worse than it was. It just means that more people have been counted. But the headline will probably read 10 times as many cases as there were yesterday, not case fatality ratio dropped tenfold. So just be prepared for that headline to happen and be really rational about it. So something else we've noticed is that it seems worse in some places than others. So there's a very high lethality in Italy and a very low lethality, for example, in Israel and Germany and some other places. So what's the difference in Italy? It's not entirely clear. So some hypotheses, some ideas that have been tossed around are, one, Italy has an older population. Italy and Japan have the oldest populations on Earth. I think their median age might be 47 compared to our 35 or something like that. And so if we know that older people are more vulnerable, it could be worse in a place that has more older people. Another issue is that they may have gotten too many cases too fast and they weren't able to test that many. And so the bottom of that fraction was too small and it looks bigger than it will be later. Other hypotheses thrown around that need to be evaluated are a very social population. It may have spread more than populations that are more inclined to be introverted and stay home. And it's also been suspected that those people with underlying tuberculosis may be more vulnerable. And we're not going to know for some time. And the way we're going to know is to go back and do serology. So there's two ways you can test for virus. You can test using PCR, polymerase chain reaction, and that looks for active viral genetic material. And that says, do you have a replicating virus right now? The other test you can do later, serology, looks for antibody. Antibody means you were infected, you mounted an immune response, you recovered. Antibody is the trace of a virus that has been there and now gone away. And so if you look for antibody in a couple of months, you'll get a sense of how many actually were infected, because a lot of them we're not seeing right now, you know, all the kids, all the young people. And you get a sense of, by looking at the antibody, how the virus transmitted. Did it really infect everybody in the household if one had it? When it got into a facility, to whom did it transmit and how and why? And what was the actual severity? And can we break that down by gender and occupation and age and health status? So that's going to be a lot of data we'll get, but we won't get it for a while. Now, another way of looking at this, and I'm not sure if this is reassuring for you or not reassuring for you, is that I'm not surprised by any bit of this. SARS-CoV-2 is just the new virus in town on top of an abundance of respiratory disease that we in the planet and in America experience every single year. So in this 1 year that coronavirus has thus far killed 20,000 people in the world, 800 in America, we have all of this disease all of the time that rapidly spreads human to human as well. So in this same year, 300,000, well, every year, 300,000 to 650,000 people die from seasonal influenza. In America, 40,000 people died from seasonal influenza between November and February. So that's 40,000 compared to a couple of hundred. So coronavirus is very small. Nobody knows what's going to happen. But every single year, even though it's a good vaccine, 140,000 people still die from measles. 1.2 million die from tuberculosis. And respiratory syncytial virus, RSV, something that kills babies and young children and also the elderly. It kills 60,000 babies every single year around the world. You know, my own kids got it and it was really scary. They turned blue and they couldn't breathe. Probably a lot of your kids got it too. Most people get it before age 5. It kills a lot of elderly people as well. I think 12,000 Americans a year or something across different ages. So this is another respiratory disease on top of what already spreads and the abundance of disease that we grapple with and attempt to prevent every single year. And so some of the public health concern is we don't want to add a new thing that could skyrocket on top of what we're already dealing with. Since we're all staying away from each other, we might get a little less flu, a little less RSV now that we're at the end of the flu season. So maybe those people will stay out of the ICU. We have 12,000 Americans every year that die from falling down the stairs. If we're all at home, maybe we will fall down the stairs more. Nobody knows. But in order to control this thing, we as humanity will need to build up herd immunity. Herd, not meaning that we're cattle in a feedlot, but that's kind of how a virus can see us. Right now, nobody is immune. This is an entirely new virus for humanity. No one has immunity to it, and that's why it spreads from one person to another. Every time a virus approaches a new person with a droplet or a fomite, it's like a roll of the dice. And it might stick and affect that person, and it might not. And every time it hits someone who is already immune, that stops that potential chain of transmission. So herd immunity of about 60 to 70% of us being immune could stop something from being a pandemic. Right now, we started at 0 at New Year's Eve, and we will, as a human race, need to build up that herd immunity, but we need to do it slowly and over time. And that's why they didn't just isolate the elderly and ill and send all the kids and teenagers out to have a giant play date dance party and get it over with, because although kids aren't suffering so much. It's, even if it's 0.002% lethal in children, someone's going to be that 0.002%. And that's not something that we find palatable as a society. And also because it's imperfect. We do not keep everybody apart. Generations live together. We cannot ever achieve perfect social isolation. We do not remain in boxes by ourselves like research mice. Even if we're social distancing, we're still going to the grocery store, we're still going to the pharmacy. We still can transmit within a household. And so we will need that herd immunity, and we will build it up, but we need to build it up more slowly. And then the question is, looking at this abundance of respiratory disease that we see every year, two things. How is that even possible? And your immune system is a remarkable thing. This is what you are grappling with and fighting off every single day. Your immune system is a remarkable, complex, powerful series of tissues and cells and organs and molecules that every single day you find and destroy viruses, bacteria, and fungi that you never knew about. You didn't get infected, you fought it off. Every single day, your immune system finds and destroys an individual cancer cell before it ever becomes a tumor or a malignancy. It is remarkable, the ability and complexity of your immune system to find and destroy all kinds of disease, whether that disease is self or non-self. And we know sometimes your immune system can be too powerful, and it can trigger something like autoimmunity, or some of the end stage disease for both flu and coronavirus is an overactive inflammation and an immune infiltration into the lungs. So it's a fascinating area of study, and it's the reason why humanity is all alive today. And there's so much we can learn and understand about how to support immunity to prevent all of the diseases, whether it's COVID-19 this year or what we're going to grapple with 6 months from now, the regular cancer, heart disease, autoimmunity that we all have. So since we're grappling with the new thing now, how bad is it going to get? Nobody knows that either. Estimates so far are that each person infects 1 to 3 other people. Virologists use a number called R naught, naught being kind of the old-fashioned description of a 0. And the R naught for SARS coronavirus is about 1.5 to 3, depending on who was in the study and where and when it was done. R naught for flu is about 1-ish. R naught for something like measles is 16 or 18. So there are viruses out there that transmit more frequently and more abundantly than this guy. This guy's about 1 to 3. But as long as it's bigger than 1, it's gonna go forward. How bad is it gonna be? Well, if we use those cruise ships as an experiment that nobody meant to have happen, but did anyway, among the humans on the cruise ship, whether they were passengers, whether they were crew, ultimately 15 to 20% of them got infected with the virus, even though they knew there was a virus on board and they were supposed to have spent that cruise this way. Now, there were some things that went wrong. They would have staff that were sick, still sharing bedrooms with staff that were well. And so infection control was certainly imperfect. But in enclosed spaces like that, 15 to 20% got infected. And it's now about 1 in 1,000 New Yorkers. So another question, will it go away in warmer weather? That's a really interesting question. So if you look at diseases with which we are familiar, so rubella, influenza, and rotavirus, you see the cyclical pattern of disease where they come and go, usually in the winter. We know that winter is flu season. That drop in rubella, that's when they introduced the vaccine, but you can still see from the unvaccinated or where the vaccine didn't take those little bumps seasonally. So if you just zoom into flu, it's flu in the northern hemisphere, comes in November and goes away in May, every single year. What's interesting is that in the southern hemisphere, where the seasons are switched from ours, their flu season is the other half of the calendar year. In Australia and Brazil, flu season is not November to May, it's April to September. It's their winter. So it's something about winter. And common cold coronaviruses have this seasonality too. So is it gonna go away in warmer weather? Well, nobody knows that either. So we can offer the definitive scientific opinion of maybe yes and maybe no. In favor of maybe yes, when the weather's warmer, people are outdoors, there's less central heating that dries up nasal passages. We will be building herd immunity, so some transmission chains will stop. But there's something fascinating about immunology here that really needs a lot more study. And that's our circadian rhythms that respond strongly to the amount of light in day, the number of hours of light in a day. The seasons strongly control not only when we wake up, when we go to sleep, when animals breed, it controls the innate variation in our immune cells that make us somehow, and for some reason, less susceptible to viruses in the summer. And they also affect, there are studies where vaccines take better if they're given in the morning than at night. So there's this one study from the Soviet Union where they gave an attenuated influenza vaccine to lots of people, and they did a study in January, and they did a study in June. So they gave the same amount of replicating viral vaccine to people in winter as in summer. In winter, they saw 7% that replicating vaccine gave them a fever, and 30 to 40% of them seroconverted. They developed an antibody. When they gave the same vaccine in June, 1% developed a fever, and only 4% developed an antibody. So that virus ramped up to a greater level in the winter than in the summer. And you might say, okay, well, you know, in the winter, they're indoors. It's Soviet Union, it's very cold, the humidity is different. So there's a follow-up study. They took research mice, and they kept the research mice in a constant temperature, constant humidity environment. And they gave them the same amount of virus. In the winter, 60% of those mice got infected. In the summer, only 30% got infected. So there was something about the seasonal patterns by which our cells and our signaling and our hormones and everything else responds. So that's in favor of maybe, yes, it'll drop a little bit and when the weather gets warmer. In the column of maybe no, that an emerging infection, something new for which nobody has any immunity, don't always follow the normal patterns. They just spread until we're done with it. So we don't know what's going to happen. Most of us are still going to be immunologically naive if we have done our social distancing well. We're going to get really sick of being alone, and we can't do this forever. At some point, we need to get back to work. And it's not clear what that point in time is, but when we start interacting with each other again, the virus will start replicating again. And the second thing is, although it might slow in the summer, it might come back in the fall. And this virus may become like other coronaviruses we see that cause those colds every winter, and you might look forward to an annual coronavirus booster vaccine. And eventually we're going to build up herd immunity so it's not such a crisis. And there's something else really interesting about immunology here, the sex-based differences in disease and in fatality. So in Italy, males were 58% of the cases and 70% of the deaths. 8% of male patients die, only 5% of female patients die. China, very similar. 64% of the deaths were male, only 36% were female. So females fare better than males with this thing. Now, some of that might be sociological. In China, about half the men smoke and only 2% of the women. So smokers could be more vulnerable to lung disease. But there's something else fundamental to our biology. Every cell of your body is either male or female, not just your sex organs, but every cell. Every cell has either 2 X chromosomes or an X and a Y. There are a lot of immune genes encoded on the X chromosome. And so females have 2 copies of those and males have 1 copy. An X is inactivated when it's an extra, but that means you might have a greater abundance and variety of those kinds of genes, which could be helpful. So here's a study from SARS-1 in 2004. And there's something really profound in which the research mice strongly reflect what was observed in the human data.
So, just to let you know, we only have a few minutes left.
My last slide.
Okay.
So, underlined in blue are things about the boys, underlined in pink are things about the girls. The male mice were definitely more susceptible to the same experimental infection. And also the degree of sex bias had to do with the age of the mice. That older male mice were more susceptible than older female mice with exacerbated disease. And it looked like it had to do with the amount of estrogen and the estrogen receptor signaling. And so some of these female hormones in these cells could be very protective against these kinds of viruses. There are a lot of vaccines in progress. It's going to be a year or 2 before they're available. I can talk about what the time length is. But what's good is that this is happening faster than we've ever seen it happen before. That usually a vaccine takes 10 to 20 years. This one I think will take 1 to 2. And they've already started the phase 1 trials simultaneously with the animal studies. They're looking at repurposing some molecules that act in broad ways. And we are leading up an effort to discover and deliver therapeutic antibody, which is a molecule I'm really excited about because it's safe and offers you effective and immediate immunity even before you've ever had a vaccine. And so that is a global collaboration that we're heading up here. And I can also tell you more about it and kind of what antibodies do. And it's a real good return on your investment. This is a couple of back of the envelope calculations. The money needed to prevent an infectious disease is a tiny fraction of the destruction that one of these causes. And that's clear now more than ever. So I'm happy to answer any questions that you have.
Thank you so much, Dr. Saphire. I'm wondering if you could spend a minute or two just talking about the work that you're doing at the La Jolla Institute in a bit more detail and about the collaboration project that you have been trying to spearhead worldwide.
Yeah, sure. We're doing two things in my lab. So we specialize in using microscopes to get very high resolution images of pandemic viruses to figure out what you look like, how you're folded up, where you're vulnerable, and where we would launch a drug, a vaccine, or an antibody. So these are not microscopes like you had in high school biology class. These are microscopes that are 11 feet tall and have a 300,000 electron volt thing. So we can see into the pieces of the virus with unprecedented, beautiful resolution, every hydrogen bond. We're using that information for a couple of things. That gives us a roadmap of how the thing works, where to launch a drug, how it is changing. So we're going to provide that fundamental basic information, the roadmap for how to design a therapeutic. Now, in the practical level, we're also using all of these molecules we make for these studies as the framework to galvanize this global study. So right now, we need to move faster as scientists. We need to work smarter as scientists. We need to combine all the tools and all the expertise and all of the brains and all the different ways that you might analyze those antibody therapeutics that could give somebody immediate immunity, whether for a frontline medical worker or a contact that's been exposed or somebody who's already been sick. We are combining those candidate molecules in different ways of developing to run a massive, rapid parallel study so that we can recommend what the best drug should be. Because when WHO says we need to pick one thing and mobilize it, what's it going to be? And one company says, I have the best, and I know because I did experiment A. Another company says, well, we have the best molecule, and we know because we did experiment B. And another group says, well, we have the best molecule, and we did experiment C. How are they going to compare? So we are running all of the experiments side by side and all of the molecules to make an independent, unbiased judge of what actually is the best therapeutic and what works. And we can use that information to evaluate the vaccines as well that will come on board later.
Thank you. That's incredible. Do you mind just putting that slide back up that shows the worldwide collection of the data that's coming to the La Jolla Institute, just so people can get a sense of that, and then I'll ask the next question. And maybe explain that a tiny bit, yeah.
So we are the headquarters in San Diego of this global collaboration that's supported by the Gates Foundation, where people making these kinds of molecules that offer immediate immunity can be evaluated and analyzed. So we are the leadership behind this program. We are also doing the molecular analysis of the molecular structure. And that molecular pipeline in a hurry is going to be really key to getting these results where they need to go.
That's amazing. Thank you. That's fantastic. We received some really, really great questions. One of which is, if the country needs to return to work and school, whether in a few weeks or a few months, but before we have a vaccine, how do you envision a model that will allow for people to go back without a reoccurrence of the coronavirus?
It's really unclear right now. I think that we need to get a good handle on what virus is out there, and then you can be rational and use a lot of common sense that people that are elderly or immunocompromised or have underlying conditions will need to remain isolated longer. As much as you want to hug your grandkids, we know that kids are germy. I got a couple of germy kids at home, and that, you know, those kids are where herd immunity is going to come from first. And that probably happened when the common cold coronaviruses first emerged into human populations 100, 800 years ago. You know, when those first came into the human populations, whether it was 1920 or 1220, no one was immune then either. We just had a real short human lifespan. And so we didn't know how lethal it was against elderly people or people with underlying complications because they wouldn't have been alive. And also nobody knew what a virus was either. There just may have been some uptick in disease and nobody knew why.
Um, we probably, unfortunately, only have time for one more question. And I think there's a lot of people who are curious about what's happening locally with a coronavirus. So actually, I'll make it a twofer. First of all, can you talk about if there is and what kind of collaboration is happening locally with San Diego scientists? And then the second part is what's going on with coronavirus in San Diego? Where are we in the curve?
Well, thankfully, it's still relatively rare in San Diego. Cases are increasing, but we're still down low. There are people in the ICU, but it's not been the severe situation that's being observed, for example, in New York City or New Orleans. One of the things in our favor is how much Southern Californians love their cars. You know, we don't pack into the subway. We drive in our individual cars. That's probably offering us some protection. And then so some of what the scientists are doing. So the SETI lab at La Jolla Institute is looking at what the immune system sees and recognizes. It's called immune epitopes. And building up large databases that we can compare across blood types and HLA types. We have a pulmonary researcher physician on staff that's looking at some of the effects of lungs. We have a researcher, Susan Shrestha, who has field sites in Nepal where the virus is first being introduced. So we can have an opportunity to observe this transmission and capacity build in a nation that may need our help. And then across San Diego, we are sharing basic research information and clinical information back and forth, UCSD, La Jolla Institute, and the other institutes in town.
Fantastic, fantastic. I mean, that's great. And it's so great to hear. And it's amazing to hear the effort that you've been coordinating and the worldwide effort that is going on to try and address this. I can't tell you how many questions we've received. It's a lot, and I wish we could ask them all, but unfortunately we only have limited time, and we also want to let you get back to the important work you're doing. So I will just say thank you once again, Dr. Saphire, for an incredible presentation, and you've given everyone a lot of to think about and to digest, and we greatly appreciate the information you presented, and more importantly, the work you are doing. So we wish you really the best of luck in your service to not just our community, but the world community.
Well, we sure appreciate your time and your help. We need it right now.
Thank you. Thank you so much. Now, before everyone hangs up, there are two very important things that you need to know about. First, the Jewish Community Foundation has set up 3 COVID-19 emergency funds to tackle different aspects of the coronavirus crisis. I'm just pulling something up on my screen. So the first is one is a public health response fund to support organizations just like the La Jolla Institute. The second one is our Vulnerable Local Populations Fund to address the multiple needs, new needs for the most vulnerable in San Diego. And the third is a fund jointly created by the Federation, the Jewish Federation, the Lichtag Foundation, and JCF for those who are most vulnerable and impacted in San Diego's Jewish community. You can get more information about those funds at the website on your screen, jcfsandiego.org/COVID-19. We are also going to be sending out an email with all the details because we realize there's a lot of information to digest. Additionally, we recorded this Zoom presentation, and so you will be able, and we're going to stick this on our website, so you'll be able to watch this later and share this with your friends, because I think Dr. Saphire shared a lot of really incredible information. And so the last thing I just want to say before we leave is this is just the first seminar in our Access to Insight series. Next Tuesday at 12, we will be having financial experts discussing the financial impact of the coronavirus for you, its implications for you, and also talking about the new stimulus package that Congress has just passed, I believe. So all this information will be sent to you via email. If for any reason we don't have your email, you can, or if you have any questions, feel free to email either Mara at JCF San Diego or me, Ollie, at jcfsandiego.org. Thank you once again to Dr. Saphire and the La Jolla Institute, and thank you all for signing in. We hope to see you again next week.